To understand the benefit of pairing an optimal dose of a medication with a medical food, let's first review neurotransmitter release and reuptake in the central nervous system. A neurotransmitter is manufactured by the neuron and stored in vesicles at the axon terminal. Vesicles release the neurotransmitter into the synaptic cleft. The neurotransmitter diffuses across the cleft and binds to receptors on the postsynaptic cell. The neurotransmitter molecules are released from the receptors and diffuse back into the synaptic cleft. The neurotransmitter is reabsorbed by the presynaptic neuron. This process is known as reuptake. For example, when the reuptake process is blocked by a pharmaceutical drug, this block in reuptake combined with a nutrient deficiency results in a patient's side effects. Why does this happen? Most disease states are associated with a functional nutrient deficiency due to the accelerated turnover rate of specific nutrients. Modern medicine has focused on treating the symptoms related to disease, ignoring the necessary metabolic requirements for neurotransmitter release and reuptake to aid in a patient's recovery. Because this cycle can be inhibited by medications, disease, malnutrition, and environmental toxins, the neuron loses its ability to produce neurotransmitters and eventually triggers cell death. As a physician, in order to prevent this from happening, your medication solution must replenish the fuel to keep the cells functioning, giving the neuron the metabolic ingredients to manage the symptoms of the disease through neurotransmitter precursors. These products are regulated by the FDA in the category of medical foods and are a safe and effective way to restore neurotransmitter levels depleted by disease. The following are some of the specific conditions for which you may now offer this solution to your patients. For example, to manage pain, compare prescribing naproxen to a metabolic precursor system called theraproxen. The patient experiences the same reduction in pain, but with the lowest FDA-approved dose of naproxen. This optimization of the dosage prevents the patient from reaching the threshold where the side effect occurs, and it does so without compromising the medication's intended effect. This graph shows a double-blind, randomized control study on the effects of theraproxen on chronic back pain compared to the effect of naproxen and Theramine alone. This study indicates that a physician can significantly reduce or eliminate the dose of an NSAID pain reliever and replace it with twice-a-day Theramine with no loss of efficacy. This study was published in the American Journal of Therapeutics in November 2010. Targeted cellular technology is a patented 5-component process that allows milligram quantities of neurotransmitter precursors to produce therapeutic effects of neurotransmitters. Previous attempts to use neurotransmitter precursors have required much larger quantities of the precursors to elicit a therapeutic effect, making it functionally impossible for a patient to ingest large gram quantities of a precursor agent on a daily basis. This is why amino acid regimens fail. The targeted cellular technology works because it targets the specific cell. This process includes a neurotransmitter precursor, an uptake stimulator, a neuron activator, an adenosine brake releaser, and an attenuation inhibitor. This patented process allows neurotransmitter release and reuptake to continue, thereby preventing the development of tolerance. And, unlike pharmaceutical agents that lose their effectiveness in a relatively short period, targeted cellular technology maintains its effectiveness and does not attenuate. The manufacturer validates its products in several ways. First, the product formulas are created using extensive peer-reviewed literature that's documented in the company's monographs and database of full-text articles. Second, it performs physiological testing on all of its products. Third, validation comes from open-label clinical outcomes trials. Finally, double-blind placebo-controlled trials are performed. Here's an example of a double-blind placebo-controlled trial for time to sleep. In this double-blind study of Gabadone, the treated group experienced a significant reduction in the time to fall asleep. In this double-blind study, the patients achieved restorative sleep. This is demonstrated by a substantial improvement in AM grogginess compared to placebo. For more information regarding medical foods, call Physician Therapeutics, a division of Targeted Medical Pharma, Inc. at 888-394-3439 or visit www.ptlcentral.com.